Laboratory technologies in immunology. This is a virtual practical lab revision. First, we'll recall about inflammation and immune response. So, following an injury, you can see there's a heart injury. So, there is a disruption of the tissue. So, epithelial layers leading to an ulcer. So, there are first cells which respond is mast cells. It's from the myeloid which release histamine and prostaglandins. So in turn, it attracts neutrophils via the capillaries. So you can see how the neutrophils reach into the tissues because neutrophils are part of our blood. There is a circulatory polymononuclear leukocytes. So they attract following these histamine and prostaglandin released from the mast cells. So first of all, it's rolling. So it's slowing the motion within the capillaries, leading to cell adhesions and activation. Rolling, or it leading to binding, but weak binding, then activation with interleukin 8. Then it leads to firm binding with the cell adhesion molecules. First, it rolling initiated with L-selectin and P-selectin, then IgG superfamily cell adhesion molecules, like which leads to firm binding, and further it leads to migration or diapedesis, transcellular migration, which enters those cells into this area. At the same time, protein may activate, leading to coagulant form. It has both advantages and disadvantages. So as a result, blood cells, phagocytes, everything comes in and it acts upon these invaded pathogens because they may be part of normal microbiota of the skin. Once heart injuries, they may reach into subcutaneous tissues and leads to skin and soft tissue infection. So as a result, these neutrophils and as well as tissue macrophages, dendritic cells, would phagocytes this and engulf and destroy it. So this is the process of phagocytosis. First, phagocytes. In the blood, main phagocytes is neutrophil. Within the tissues, macrophages. So first of all, there is a identification. Then they would come and adhere to the cell membrane attachment. Then they form this phagosome or pseudopods. Pseudopods lead into engulf, taken into. Then it's form the phagosome. Then lysosome and phagosome fusion. So then within that, they destroy these pathogens. They are byproducts or debris from the cells, dying cells, with the help of reactive oxygen-free radicals and reactive nitrogen species. Reactive oxygen-free radicals like superoxide, hydrogen peroxide, OH dot hydroxyl moiety, or nitrous oxide, peroxynitrates, like that is from the NO plus superoxide. Then if there is a myeloperoxidase activities, they perform HOCA, hypochlorite acid. Likewise, they leads to oxidative burst. And ultimately, these debris will be removed out. So this assay called nitrobi tetrasodium assay. It is done for assess the reactive oxygen species detection. So NBT assay, it detects the activity of NADPH oxidase enzyme, which converts or form superoxide, hydrogen peroxide, and hydroxyl moieties. So when they are able to form, so this NBT assay is a chemical used among these neutrophils and other cells, and with the presence of NADPH2 activity, which converts into the yellow into blue color. So in the normal cells, we can detect NADPH activity. And induced cells following inflammation, we can detect much more color change into blue is much high in frequency. So if there's NADPH oxidase deficiency, there's unable to produce those reactive oxygen species. Ultimately seen as yellow, no color change, NBT negative. So there are instances we can see mixed populations, maybe the positive or negative in mix. So we can look through the microscope and we can appreciate the color change, and intensity of color change we can detect by using absorbers uh, by detecting the known nanometer frequency. So based on that we can detect intensity of the color change as well. So then it can be quantitative as well. Early it may be positive, negative, qualitative, and levels we can detect by absorption. Then it is quantitative as well. Bit of pro-inflammatory markers. So interleukin 6 is the interleukin 1, TNF alpha are considered as pro-inflammatory markers, which mainly secreted by mast cells, neutrophils, even macrophages for the inflammation response. Maybe following trauma, following any microbial invasion as a first-line defense in innate immunity. So those IL-6 which comes to act on IL-6 receptor on cell surfaces. Okay, then it leads to angiogenesis, proliferation, likewise process. This is a cell signaling pathway downwards within the cytosol. This is the membrane, this is the extracellular part. Similarly, TNF alpha also acts in the same manner upon different functions. Those are termed as cytokines, what you learned during the lectures. So how to detect cytokines? There's a method called ELISpot. So it is used to measure the frequency of cytokine secreting cells, T or B cells, at the single cell level in response to specific antigens. So we stimulate using known antigen. Then known antibodies are quoted and we try to figure out what are the secreting cytokines. So then we can quantify. This is called capture antibody. These are the cytokines. Then some cells. So and detection antibodies. Likewise, we may able to detect at like we can see like spots. That's why it's termed as ELISpot. Interferon gamma ELISpot for T cell response. B cell ELISpot, antibody secreting cells. You know B cells. Dual color ELISpot. Likewise, fluoro ELISpot. There are several methods. So ELISpot is for the cytokine measurement. So acute phase response. So it's a defense or inflammatory response against invading pathogens. So it is mainly driven by interleukin 6, which goes and act upon the liver and produces positive and negative products. These are positive products, increase C-reactive protein, serum amyloid, fibronectin, ferritin, likewise. Whereas there are some products, it decreases. Factor 7 albumin alpha protein transferase. So each has different functions. So at least if you know what are the functions of C-reactive protein, which goes and binds to bacterial phosphocholine and enhance phagocytosis by opsonine activity, as well as C-reactive proteins are capable to activate complement system. Serum amyloid, there are different functions. Likewise, you may have to see those. And albumin reduces because it allows to produce other protein fractions. Transferrin, which is iron transferring protein, which reduces because iron is required for some bacteria as a siderophore activity. So that's how those C-reactive proteins acts. So very much important for the first line defense, which protects body from those invading pathogens. You can see how these electrophoresis, how the albumin looks, alpha 1, alpha 2, beta 1, beta 2, and gamma assays. So gamma is the immunoglobulin fraction within there. So acute phase response changed much to reducing the albumin while increasing alpha 1 and alpha 2 components. Immunoglobulin also stays further, maybe rising later in the time, but this is the response alpha 1 alpha 2, while albumin may reduces in the concentration. CRP detection. So it's often done at the hospital setup because C-reactive protein. So we use as an inflammatory marker. So we can do either using the manual methods, using the card assays and visual agglutination method. So that is, we put patient serum and we put the anti-CRP antibodies containing reagent and we make a serial dilution and try to find what is the exact concentration. But there are other methods like automated methods. So with the help of fluorescent label antibodies, we are able to detect those CRP levels in milligram per deciliter in exact value. Not like here I told exact, but it's depend on the concentration. It may be 6, 12, 24, 48, 96, 192 like. But here we can see detect exact value, maybe zero point 0.5, maybe 5, maybe 10.3, likewise in milligrams. Other thing is about the complement assays. You know, complement system, it's part of innate immune system, but it may activate part of adaptive immune system like following classical pathway because it requires immune complexes to activate antibodies, while other two pathway, alternative pathway and mannose binding lectin pathways are part of innate immune mechanisms. Ultimately, they produce products like C3A, C5A, C3B, C4B, C5B, then C5B to C9 complex, making membrane attack complex. So we are able to detect those as AD and CD, like that is alternative pathway AH. CH is the classical pathway. There are ways to detect the uh, classical pathway activities, alternative pathway activities, final common pathway activities, and even individual levels of C1 to C9. And also there are products like property factor, I inhibitor factor, protectin like CD59 factor for MAC. There are different factors. This is about the allergen detection. So you learned hypersensitivity part of immune response. So there are certain moieties which can evoke immune response and lead into allergy among some individuals depending on their genetic vulnerability. So we put their skin prick test and patch test. So and also we may able to detect specific IgE from the blood. So skin prick test is we put allergen solution placed on the skin and then we put a needle and prick in the intradermally. So then we keep it for a certain period, maybe 48 hours, but then we see is there any reaction. Positive test skin is red and itchy, so it's provoke inflammation. You can see those are positive while others are negative. This may mean duplicates. No, it's a different positive and negative reactions. So it's a skin prick test. While patch test is we put these patches, non-patches, and see allow certain time and see whether is there any immune response or inflammation. So about the penicillin allergy. Penicillin undergo metabolic transformation following injection or after intake. It form penicilloyl protein, penicilloyl mainly, and then it conjugated protein, leading to protein conjugate. So that is the major component. So it's a penicilloyl. Let's see, count for 95% of penicillin bound protein form the major allergen antigenic determinant. While there are like penicilloid, penicilloid, and benzyl penicillins are minor components, also provoke allergies. So penicillin acts as a heptane, then bound to the protein carrier molecule, then it becomes heptane carrier complex and it provoke immunogenicity. So how to detect penicillin allergy? So as no primarily cause type 1 hypersensitivity mediated by IgE antibodies. So when penicillin and they are metabolized by host protein, they form peptide carrier complexes are recognized by the immune system. So those who are sensitized, these complexes trigger B cell activation, IL-4, 5, and then products the specific IgE, which binds to mast cell basophils, which release uh, histamine, serotonin, and leads to immune reactions. So that is degranulation, release histamine and other mediators, caused in urticaria, itching, angioedema, severe form, and most severe form, anaphylaxis. Meanwhile, penicillin allergy can be responsible for type 2, type 3, and type 4 abscesses as well. Those are non-IgE mediated reactions, like serum sickness, that is type 3, then hemolytic anemia, type 2, or delayed rashes. So how to detect? We put penicillin, no, they are byproduct, major component, penicilloin. Then we put the saline as a negative control and histamine as positive control. And we allow to react certain time. And then here it is negative. Histamine, yes, it's a positive control reactant, but penicillin, this patient is not a sensitized person. So it is negative. If there's a positive or sensitized person, patient or person, there is a reaction. So then the paramount, most important aspect of immune-mediated diagnostics and applications are antigen-antibody reactions or interaction. So we use antigen-antibody complexes formation to detect antigens from those pathogens or tumor markers or antibodies for react against those invading pathogens to detect like in dengue and even hepatitis like chronic infections as well as for those about the autoimmune reactions. So we use the concept of precipitation, that is visible reactions in the known antigen and antibody bounds. It form a complex and it may precipitate or agglutination test. So that is the epitope of antibody binding region. This is the antigen determinants and binding all the variable heavy chain and variable light chain areas, that is termed as paratom. Once this antigen and antibody bonds and multiply bound for another one, like was here two arms, another two arms, so they form a complex. So it is termed as immune complex formation. So in diagnostic immunology, we use what I told earlier, antigen antibody detection. So there are clinical sensitivity. So that is ability of test to provide positive results if the patient has the disease. While specificity is ability of the test to give negative results if the patient does not have disease. So no false positives. So those test validity becomes on the in the sensitivity and specificity is higher. To develop those known antibodies, there is a technology called hybridoma technology. Concept is that imagine we are going to detect an antigen. We'll say dengue NS1 antigen. So to detect that, our tool is anti-NS1 antibodies. So we have to produce those anti-NS1 antibodies in a large amount. Those are termed one is monoclonal antibody because it is specific for that NS1 area. So those known antibodies are capture antibodies are important because that is our tool in this case to detect the whatever the antigen. So think there are a lot of antigens. So to detect those different antigens among different diseases, HIV, dengue, and also certain tumors, even for the inflammatory markers, likewise. So we use different technologies like enzyme-linked immunosorbent assay, radio immunoassays for all the things which those plates contain wells. Within the wells, antibodies should be there to detect. So those are human made ones. So manufacturing in the mega scale. So we need a lot of antibodies to put them in the solid phase of their those wells. So we have to produce those. For that, one technology is we use multiple myeloma cells, tumor cells, and uh, adjoin with known plasma cells which are able to produce, in this case, NS1 antibodies. So this is immortal. These cells are immortal. So there is no any death. They produce continuously. These are mortal cells, but they form hybridoma cells. It's a hybrid. So it's able to produce non-antibody and also it is immortal. While tumor cells doesn't produce antibodies, but it is immortal. So this is mortal. Once they continuously produce antibodies, whatever we require. So we may use rats or cell culture techniques or humanized antibodies, that is human produced and put into the poor mouse and mice, and we allow them to produce those known antibodies. So those we use for different aspects in medicine. For like infectious diseases, we use monoclonal antibodies to treat toxic shock syndromes, toxin associated condition, and also hepatitis B immunoglobulins, those who have recently having a prick injury following non-hepatitis B positive patient, maybe healthcare worker, maybe drug user, likewise. So as the immediate measure of passive immunization, we give those non-produced immunoglobulins. Even for the tetanus, botulinum toxin, for the again botutoxin diseases, likewise, there are a lot of monoclonal antibodies are available. And autoimmune diseases, yes, rituximab, likewise, also maybe anti-cancer therapy. We are targeting non-receptor, likewise, producing those antibodies. Then for certain allergic conditions, immunotherapy, yes. Then neurological disorders, cardiovascular, transplantation, bone marrow, likewise, there are a lot. Bit about daily diagnostics. Now you know about the antibodies, about IgM, IgG, and this is the primary response. If you recall, primary response, innate response is all the same, no change. But initially it is secreted by IgM, then later IgG. But when the secondary exposure, prominent one is IgG antibodies, and IgM also synthesizes a little. Sorry. So what we do is we use a lateral flow immunochromatography assay. So this is a cross section. So it contains this area antibodies conjugated with gold, latex or fluorophore, depending on them, allow to capillary flow. Then test line. Known antibodies are there. Control line. Again, IgG antibodies are there. So we put the sample there, analyze, then allow certain time to flight or flow over this capillary flow. This, if there are known antigen, antibody will bind in the conjugate pad and along this nitrocellulose membrane, which migrate and bound with known antibodies test line, as well as control line, positive, and control should be there to be positive, so it is a valid test. So you can see here, uh, this first of all we have to see this lateral flow immunochromatography assay. First we have to see control has been positive or negative. Here both, but test it seems it's negative, but it is here test positive and it gives both Ig, here it's IgG only and control positive. So here also control positive, test negative, likewise. So there are other fluorescent antibody tests. We use direct immunofluorescent assays like fluorescent antibody testing. Detect localized antigen in the patient. You can see how the things going. So non-fluorochrome antibodies, they are tissue section list from antigen. Antibody to tissue antigen is labeled with the fluorochrome. That's how it goes. So it bound and emit immunofluorescence. So direct immunofluorescence, group A, for example, so those are known as strep. Then fluorescent dye-label antibodies to group A. So they bound and mount and immunofluorescence, emit fluorescence. Indirect immunofluorescence is used to detect pathogen-specific antibodies in the patient. We can use antigen detection as well, but more often we do for antibody detection. So we use laboratory generated sample of infected tissues mixed with the serum from the patient. Then the fluorescent dye-label anti-immunoglobulin is then added, maybe raised in animals. If binding of the antibodies from the patient to the tissue samples occurs, then the fluorescent antibodies can be bound and fluorescence can be detected in the tissues by fluorescent microscope. So we use for the detection of anti-Epstein-Barr virus capsid antibodies, anti-double-stranded DNA antibodies, anti-thyroid antibodies, anti-nuclear antibodies in SLE. Here it is Hashimoto's thyroiditis. This is maybe SLE. Anti-glomerular basement membrane antibodies like this. So here, unlabeled antibody is a known antigen. We want to detect unlabeled antibody and fluorochrome labeled anti-immunoglobulin against this. This is an antibody against for the antibody. So then it's that's why it's termed as indirect. Then fluorochrome label one we use for detect one. So it here Trichodoma pallidum from laboratory stock. Specific antibodies in the serum of the patient. Antibody binding tripallitum. Fluorescent dye-label anti-human immune serum globulin. Fluorescent spirochetes. In suspected Graves' disease, the first line test is antibodies against THS receptor, which occur with the prevalence of 90 to 100%. Here we use tyrosine peroxidase. Yes, tyrosine peroxidase can support the diagnosis of ELISA. So this for the Hashimoto's thyroiditis, like serological antibodies against TPO are detected with the prevalence of 90%. Then there's another concept is radioimmunoassays and enzyme-linked immunosorbent assays. So RIN and ELISA are extremely sensitive tests that are common in medical laboratories. Both tests are conducted similarly, but RIA uses the detection of radiolabeled products and ELISA detects the presence of enzyme-mediated color changes in a chromogenic substrate. Just simple, just don't go much, but so we use the radiolabeled emission. When the antibody and antigen complex are being there, then it goes and there's a competitive actions between radiolabeled antigen and the non-antigen, and it's a non-antigen goes and bounds with there, and unbound fraction is much, and it's any immune or detection ability. While ELISA, same principle, but we use here detection of P24 antigen in HIV and we produce P24 antibody which bounds and then we give another antibody anti-human immunoglobulin. It contains the enzyme portion. Once this anti-human antibody bounded, it leads to activation of this enzyme portion and leads to production of the chromogen, converts into the color emitting product. So it's a quantitative assay, qualitative as well. Quantitatively based on that with the serial dilution, we can detect the exact amount. So here you can see up to 4, here up to 1 only, here up to 6 positive. These are the positive control, these are the negative controls, microtetraplate. That's how it detects. Then there are different methods. We use double antibody ELISA. That is, we use another antibody. First, we put in a solid phase that is within those wells like here, same different wells. Then we known antibody is there. Then we put the patient sample. So antigen will be immobilized because antigen antibody reaction. Then we put another label antibody. So it's like a sandwich. It's a quantitative version. So ELISA we use to detect antigen antibodies for HIV, West Nile, dengue, hepatitis B, hepatitis C, lot of infections to detect and also assess the response. And also for food industry detection for food allergens, milk, peanut, walnut, almond, and eggs, we use those. Then that's the other important thing is about the fluorescence activated cell sorter, or it is termed as flow cytometer. This procedure is used to rapidly analyze cell types in a complex mixture, because if in the blood it's a complex mixture, and sort them into different populations based on their binding to specific fluorescent dyes. By using antibodies against specific cell surface markers conjugated to different fluorescent dyes, it is response or possible to analyze the relative number of cells present in specific tissue location. Further, if you go, as cells pass through the apparatus in a single file, the computer-generated graph is produced. So that's how. So you can see known cell surface marker with anti-CD8, anti-CD4, anti-CD3. CD3 is there, we produce known antibody and allow to scatter. Then one cell at a time allow to pass. Cells with fluorescent antibody because it been caught. Then fluorescent detector with the laser beam, it quantified it. And also you are able to separate. So it is flow cytometry. In the suspension are labeled with fluorescent type, direct or indirect fluorescent. Then we have the same flow cytometry. So you can see how the cells are labeled with the appropriate fluorescent antibodies. Cells in suspension are passed through the machine in single file in a stream of fluid. Stream is focused to one or more laser beams, measuring light scatter and fluorescent characteristic. Fluorescence are detected by photomultiplier tubes. Signal are sent to computer for analysis. So we may use only one parameter, only one marker, or sometimes we may able to use two markers. We elicit one for red fluorescent. We'll say we are going to detect CD4 cell. So CD4 cell contains CD3 as well as CD4. CD3 is part of their T cell receptor. So CD4 is, it's a part of again T cell receptor. So we put anti-CD4 as well as anti-CD3 antibodies. Then with the help by those two parameters, we are strictly able to detect CD4 helper T cells. Similarly, if you put CD3 plus CD8, it detects cytotoxic T cells. Then there are other further we can enhance by adding four markers, six markers, like four, to detect one specific cell. So that's the advancement of flow cytometry. So we may able to detect myeloma cells likewise. So there are common applications: antigen specificity, cell proliferation, cell sorting, cytotoxic assays, phagocytosis assays. Likewise, we can do a lot for detection. So that's a very important test. It's not much readily available. So thank you very much.